Thank you for tuning in to watch this interview. My name is Christopher Uhde, Pharma and Biotech Analyst at SEB, and I'm very pleased to welcome FluoGuide's Management CEO Morten Albrechtsen and CSO and founder Andreas Kjær. Welcome, and thanks for coming.
Thank you for the introduction, Christopher.
So perhaps why don't you give us an overview of FluoGuide before we dive into the Q&A?
Well, we'll be pleased to. We have a few slides just giving a summary of where we are and our outlook, and then we're very pleased to have the discussion afterwards. So just summarizing what we are doing, I mean, in FluoGuide, we help precision surgery, and we do that for cancer patients. And what we do basically is lighting up the cancer so the surgeon can see the cancer and remove all cancer the first time. The way it works is that we have a compound that is a freeze-dried powder, is reconstituted prior to use. It will be injected into the vein of the patient prior to surgery. Then it will circulate in the body. It will bound to cancer, specifically to cancer. And then during the surgery, the surgeon can switch on the light and they can see the cancer. So that is in very essence what we're doing. And if you take the next slide, the key element in our strategy is that when we say bound to cancer, it's a UPAR target, and it means that it's a enzyme that is bind to the surface of the cancer cells that is responsible for breaking down the normal tissue so the cancer can invade into the tissue. And that means that it's a target that is expressed across all solid stage cancer. And that means that we, in principle, can treat any or help surgeons in any cancer that remove that is a solid cancer. Basically, I think 80% of them have it extensively expressed. So what we want to do is that we want to help as many patients as we can. And the first indication we have targeted is aggressive brain cancer. That's a 15,000 patients in US per year. But if you look at the entire target population, it's about 3.5 million patients annually. And we will like to increase the number to help as many patients as possible. Then there's another aspect of our molecule, and that is that it can actually, when we light on it, it heat up, and it means that it can destroy the cells it bound to, and it's termed the photothermal therapy, and that's one of the things we are going to discuss today. And what we want to do with that is that we help each patient the most, and that is that we can take it from not only guiding the surgery and removing all the cancer, but we can actually remove any cancer that should be left after surgery. And then the last point is that it's not just enough to get the product to the market. We also want to have it penetrated into the market quickly. And that is why we're working on the commercial fit of the product. So it's fitting with the time of administration. We work with partnering in head and neck to accelerate the commercialization. And that means that we can help as many patients as fast as possible also. And if you take the next slide, then that is what we do, and then is what you can expect of us in the near term. So we have proven that our product have a very strong result, proof of concept in head and neck, aggressive brain cancer, and lung cancer. It's proven to be very well tolerated, and also that it works. We have proven on a regulatory endpoint the completeness of surgery, that it works in aggressive brain cancer. We have had orphan drug designation assigned in the US for aggressive brain cancer, high-grade glioma. We have received a grant on photothermal therapy. And with all that, we are now evaluating the potential of photothermal therapy in patients. We are looking into a phase 2 trials in head and neck, expanding the partnering and commercialization opportunities. And we have ongoing, of course, regulatory consultants. You should expect the first partner deal we do in 24. When we say this, I think it's important that this will be more validation than cash upfront, but it's important part of our strategy. In 25, we will have the first result, clinical result in photothermal therapy. We will have the result on the head and neck. We will have initiate the registration trials in FG001 in aggressive brain cancer. And then you should expect more partner deals to be added on top of it. And hereby, a very brief introduction to what we do and what you could expect to see in the future. And we are very happy to discuss a question from you, Christopher.
Okay, great. That's very clear and run through, and I think really exciting in terms of the amount of news flow you have. I think that's one of the things I like about your story is how I mean, quickly things move compared to regular drug development companies. But I guess the first question before I think because some of the new things to talk about are really about the photothermal therapy implications, what that will mean sort of for the program going forward, and then the design of the program, of course. But maybe just before we dive into all that, we kind of take a step back and talk a little bit first about the platform. So I think in general with fluorescent guided surgery, we've seen sort of increasing interest from drug contrast agent developers. And obviously there are a number of drugs that are on the market or in development for this. So how does FG001 and your platform stand out compared to those? Why choose this platform?
Yeah, maybe I can start a little bit to elaborate on that. I think it discriminates itself in several ways. First of all, it's going for UPAR, which is a target that is expressed almost in any solid tumor, typically 80-90% of cancers of different solid types. So it means that once we develop the product, it's not locked into a single indication. So it's cancer specific, but not cancer type specific. It's cancer type agnostic. And that, of course, gives us many opportunities, but we also have limited bandwidth. So we have to position it where the unmet need is largest, where it's best used. But again, it's the development of the drug is only once, and then it works across. Then it works, and it's a little bit depending on which of the competitors you are looking at. But this is the broad application. Then, of course, it works in the fluorescence. It works with is in the near infrared wavelength. And what does this mean? It means that it penetrates quite well into the tissue. So in the brain cancer case where we compare with 5-ALA that is already out there, we look 1 to 2 centimeters into the tissue, whereas 5-ALA is 1 to 2 millimeters. So for the surgeon, this means that as soon as the surgeon is close to the surface, the surgeon have a kind of pseudo 3D impression of what to expect and where it is. So it makes it much easier to work with. Yet it's not a totally new thing, ICG, the fluorophore we use. And this also means it's a good match for the majority of equipment that is out there. And of course, ideally, we also can be used broadly across different platforms. And then finally, and I guess that's maybe with our recent news flow, some of the more interesting thing, we have the ability to add photothermal therapy on top of that. And why is this? This is because ICG not only lights up if near infrared or if a laser in the right wavelength is lighting at it, then it emits light. But if you apply a higher energy, it actually converts this energy into heat. And in this way, we can heat cancer cells to a level where they die without heating normal tissue. And we have strong proof of concept of that in preclinical models, in animal models of human tumors. And this, of course, is interesting because it's the same compound. It's the same compound where you could ideally do surgery as a step one and then do photothermal therapy. So you have a two-step shop in a one-stop shop. So this, of course, is quite ideal and is exactly what we are pursuing right now. And then maybe finally, without making the answer too long, we also have a very favorable what is called pharmacokinetic profile, because we have a very broad window where it can be administered, typically the same day in the morning, but more convenient for the surgeons, as we are told, anytime the day before the surgery. And why is this important? It's important because very often surgeries are rescheduled. There might be a more acute patient that needs to be operated first, and you are not locked up into a narrow window. If we again maybe compare with 5-ALA in the brain cancer case, that needs to be administered 4 to 6 hours prior to surgery. So if you give that early in the morning, by the way, also sometimes a little bit inconvenient if you are operated 8 o'clock and you can do your own calculation when you need to administer the compound. Then if another patient is pushed in before and everything gets delayed to the afternoon, then you're actually outside the window where it can be used. And we are not under this restriction because there's a very broad window. So I think a lot of things that discriminates our technology from some of the other competing technologies.
Mm-hmm. And I guess fair to say also the ICG makes it a little bit less or sort of a little biologically de-risked as well, right? And then you have also the fact that it works with a lot of different equipment too, right?
Yeah.
Correct.
And then in terms of the therapeutic areas, so you mentioned glioblastoma, you mentioned head and neck. You've looked at a number of others already in terms of demonstrating presence of UPAR and the detection. How big are these opportunities, and what are the principles that determine which ones you've selected to go first?
Yeah, I can continue a little bit, and then Morten can on a more commercial. I mean, it's all about unmet need and adoption. So you could take these in theory where it will work across different solid cancer forms, but where the unmet need may be different. There are areas where surgery on outcome is more successful, and there are cancers where it's more tricky to get the right delineation of the tumor. So this is one aspect, the unmet need. So really helping the patient where it's most important. That is one thing. And the other thing, of course, is also how much is it into the workflow? Is there already used equipment, let's say microscope or surgical robots that either already have or can easily be equipped with near-infrared vision, because you need this special vision, and therefore it's an easier adoption. Let's say brain cancer, where 5-ALA, coming back to that, is used not by all, but some surgeons. So the idea of being guided by an optical signal is easily bought by the neurosurgeons. There might be other areas where it has never been applied, and therefore it's a little bit of an adoption curve and a little bit of convincing that, well, maybe it's not always so good as you believe, and therefore you need optical guidance. So I think that's really what has a lot to say, and they are let's say the two lead indications right now where we are moving forward in brain cancer and head and neck cancer. They are particularly well positioned for this in head and neck. It's known that it's all about obtaining so-called negative margins. So you need to know that when you take it out, your tumor, you can measure pathologically how close were you, and that directly relates to your outcome. And you don't just need to remove the tumor, but it has been proven that you need to move an additional 3 to 5 millimeters to obtain a good result. The so-called close margins, where you remove the cancer when you look at it in the microscope, but you were very close. That gives you a poorer prognosis than if you have 3 to 5 millimeters of margin. And thereby it's when we talk with key opinion leaders and present and discuss at conferences, then they buy into this concept. Yes, we want to become better at obtaining these margins. So that's one thing. And I already talked about the brain cancer, where it's so difficult to remove everything. That guidance is not only needed, but they are also used to using 5-ADA. So that is really what we. But we're keeping the other things in the background, and we, as you know, we have data also generated in lung cancer. So this is one area, but of course in the future we're likely to produce data in other areas, and you know, by chance, even in meningioma and other brain cancer, we had a case where it wasn't known that it was a meningioma patient, and it also worked there.
Yeah. And so I don't know, Morten, did you have any comments on the commercial side of the TA selection?
No, I mean, I think it was just on adaptation. I think that was on the equipment, as Andreas slightly alluded to, is a quite important one. And we see that it's a major things to be aware of a company of our kind. We see that most other companies have had problems with the penetration into the market due to some kind of too tight relationship to the wrong equipment manufacturer. And we see that as a key element into it. And so, as you said, the ICG is very, very nice that we use it, because all equipment that can detect ICG can also detect FG001. So in that sense, flexibility toward equipment manufacturers is quite important.
Yeah, and then so now you brought up photothermal therapy earlier, and I guess you also obviously have the fluorescence guided surgery specific use. What can you tell us about the pricing implications of the one versus the other, or having both?
Mm-hmm. Yeah, I mean, I can start here. So the price for a fluorescence-guided surgery product, I mean, if we look at the other product that's been launched into the market in the US, is approximately $5,000 per treatment, roughly in that area. And the price for photothermal therapy, we see one in Japan that was launched at $150,000 per treatment, and they have then reduced it as we go on, but still $75,000 per treatment. So a significant increase compared to the fluorescence-guided surgery price. So what we see in terms of, I mean, this of course is one aspect that makes it interesting for us to look at photothermal therapy at this stage. But also it's important to bear in mind how can we price differentiate between the two when going to the market. And the good thing here is that we can still sell the compound for the fluorescence-guided surgery price, and then we can charge on use of the equipment for the photothermal therapy on top of that. So that is a way where we can actually price differentiate and be able to both have the fluorescence-guided surgery price for the fluorescence-guided surgery applications, and then have the photothermal therapy price for the photothermal therapy applications. So it's a very neat thing that is very important as well.
Okay, that's interesting. It's also a little bit, it's new from what what we've discussed in the past. So that's how easy is it to actually secure that? How can you guarantee that? I mean, because they're already using the equipment. How can you guarantee that they can use, that they can pay, that they will have to pay the price that you want for the photothermal therapy, rather than just finding some workaround? Is that?
Yeah, I mean, as you go from an imaging system to an treatment laser system, you have a higher regulatory requirement. So the two will be combined when you have photothermal therapy, and that means that you would not be allowed, or it could be potentially unethical or dangerous to use a non-approved laser system to activate it, because it will be adjusted, or it will have to be calibrated together. So one of the key things we're doing in this coming year here, or this year, 24, is optimizing the laser to the drug, and the drug to the laser. And that means, on the other hand, on the flip side, that the two are closer bound to each other, and also mean that you have to use them together when doing the treatment. So this is kind of fit quite nicely together here.
Yeah. And I guess it makes it more of an attractive offering to a potential partner, but maybe we should come back to that in a little bit. So why don't you tell us about the data that you've seen so far across tumor types in the clinic?
Mm-hmm.
Yeah. So in general, on the optical guidance, not the phototherapy, because there we are preclinically. Yeah.
Indeed.
So basically, we have seen, just to take a little bit all the studies we have done. Of course, we started out with finding the right dose. You are cautious and you start low and you move up, and there we could see that we had very nice signals from 8 milligrams up to 48, and we then chose 36 in brain cancer milligram as our dose. You go up as long as it gets better, and then you don't take the last step where it didn't get better. So this made the dose selection of 36 milligrams. All these were basically, all these tumors were positive. It also tells us that we have a very wide range. If you can see it with 8 milligram and you use 36, it's not a question of a little bit of a heavier patient that in principle gets a relatively seen lower dose. You are not having a narrow window. And of course, it was well tolerated, clearly fewer related side effects than 5-ALA, that is hampered by some degree of nausea and vomiting. So this was the first, and we could see the signal. Then we moved on to, in the first study that we did, meticulous sampling around the tumor in the brain. We sampled in areas that lighted up and in areas that didn't light up. So in areas where FG001 told us this should be cancer, and in areas where FG001 was telling us this is not cancer because it's not lighting up. And we had 31 samples across the patients, and it showed us a very strong data on that we have a good match, what is called accuracy. In this particular study, the patient also were given 5-ALA, so that they actually had both. And since you use different light or filters to see the two, you could actually look with one set of eyes and another set of eyes, and we could see the match. And basically, we showed that we are at least on par with 5-ALA in accuracy. You want to know whether it's correct. Mm-hmm. So this was the first part. And then when we moved into the next study, where patients were not receiving both 5-ALA and FG001, but FG001 or 5-ALA, so they were what is called randomized to these two groups. And what we did there was doing it like the surgeon would do it, so in white light, so without using FG001 signal, the patient was operated on until the surgeon said, now I removed everything that I identify as cancer. And then the near-infrared light was turned on, so one could see the optically labeled tissue. And then in 12 out of 12 patients, because this was a study of 24 patients, 12 in each group, then the surgeon said, oh, there's still something left that lighted up. And that was in all patients, in 12 out of 12 patients. And then this was removed, and it was sent to pathology to confirm that it was actually cancer. If you remove something that was false positive, it wouldn't have helped you. So basically, that study showed that in every single case, you removed additional true cancer that you hadn't detected in white light. So this is basically the brain cancer part, and these data we interacted with FDA on to get kind of their feedback on how we were considering going forward. We can return to an outcome of that, which was very positive, but they were packaged. So we think we have an extremely strong case. It works, it lights up with high accuracy on the cancer, and it aids the surgeon because the surgeon in all cases finds extra cancer. Mm-hmm. So that is basically where we are in brain cancer, aggressive brain cancer, I should say, and where we now are exploring how we can integrate the photothermal therapy, because there's really a need for this in brain cancer, because it's so difficult to get everything removed.
Right.
By the way, there are actually two ways we can position photothermal therapy in brain cancer. And one is when you have removed the tumor and there is the cavity and you have some seed of cancer at the edges, that is where you can apply the laser and kind of burn away what you missed. And other positioning is, of course, that in some cases, the tumor cannot be removed entirely because it's sitting in areas that are critical to remove. Or where you would have something, you might have big vessels that you don't want to do a lot of cutting in that area. And there it may also be accessible to try to remove these parts in your strategy of surgery with heating it up, burning it away, because it's more gentle, it won't start bleeding, instead of the other, instead of doing it with knives and with suction. So a little bit of different ways that it can be positioned. Maybe the first part is the more obvious way to do it, but definitely a tool that key opinion leaders buy into both of these scenarios. And that's in general. We do a lot of interaction with key opinion leaders, go to the big conferences where we set up meetings with them, but also one-to-one on a lot of meetings. And basically, I must say, there's a lot of support and buy-in to the path we are taking. And this includes adding the photothermal therapy on top of that in this particular indication.
And it, of course, changes the risk-benefit equation as well, with all, a range of borderline cases. So I'm sure there's a lot of benefit for strategy too as well.
Yeah, and there are, and of course, what we have looked up, and now maybe that should also be mentioned without going too much into technical details, but we have looked at primary aggressive brain cancer. So the first time surgery is done, but unfortunately they relapse, they come back, and that's a whole area of its own. Doing surgery on something that came back, and of course something we also will look into. Since they all come back, you could imagine that the case mix is always almost 1-to-1, unfortunately, because everything you do surgery on is also needing an extra. Then you at a certain you don't continue just doing the surgery.
Yeah.
In the head and neck cancer, we also have very nice data. We did a new dose escalation because in theory, there's a blood-brain barrier. It might be that you can dose lower, and we have indications that you can actually do this in these cancers. They all lighted up, and both during surgery, looking at it, and also taking out the sample. So we have some strong indications based on this that the idea that you check for these negative margins, whether you are sure that you had a certain amount away from the border of the cancer, is a good way to go. And this, of course, will be done on the specimen side, because if you remove all cancer, but very close to where it stopped, you would still not find anything left in the cavity, but you want to look also at what you removed on the back table and check how close you were. So basically, this is a real-time pathology you could do. And this is extremely important, as we have been told discussing with the key opinion leaders, because what happens today is that normally the information about the close margins, or even the positive margins that you didn't remove everything, that you don't get that during surgery. So you get that late. And then, on average, you are not doing repeated surgery.
Sure.
Then you add chemotherapy, radiation therapy, a lot of side effects of that. Whereas if we can do the pathology while at the operating theater, while the surgeon is still there with the patient on the bed, then the feedback can be given immediately. In this area, you left something behind. They can remove it, and then you obtain a much better prognosis. Yep. So again, a concept that 9 out of 10 are buying heavily into. Yep. And we will make sure that the last one will also understand it.
Yeah. Now, I think there's something that came up when the previous time I talked with you, Morten, about the photothermal therapy angle and FG001, because there's, of course, another way of zapping cancer cells with contrast agent, which is photodynamic therapy. Now, you mentioned that there would be some element of this photodynamic activity, so basically free radical production as a result of activation of the ICG. To what extent do you think that might have a meaningful contribution to, that you might end up seeing a sort of a synergistic effect as compared to, say, if you only had the one or the other? Is that anything that is worth contemplating?
You want me to take it on?
Yeah, I think you take it as well.
No, yes. Definitely. I think it's a little bit by chance that we named it photothermal therapy, because that's where we have strong data and we can see it heat up. But all theories and also studies, we're actually moving studies with the funding we got on this photothermal therapy. We are establishing and collaborating on assays that can actually look at these free radicals. And I personally do not doubt that it's a mix of the two, and hopefully, as you also indicated, a synergistic effect, so that you might see something even with lower temperatures. But I don't think there's any doubt that you also have the photodynamic therapy.
Right. Okay. Super interesting. Okay, so let's talk about then the implications of trying to go for photothermal therapy in terms of how that makes you design a trial program. And I guess your thinking's evolved a little bit on this sort of in light of the phase 2b data. And so maybe you can start about I'll leave it up to you, but perhaps start about talking about the FDA view of since you've had an interaction with them, about what the best way to go about developing FG001 for photothermal therapy might be. Yeah.
If I start, then you continue, Andreas, if that's okay. So in short, I mean, the FDA, what they gave us of feedback were that the program for fluorescence-guided surgery, that they would like to see in 6 months and 12 months follow-up as well on that treatment. And that means that the program that we run for fluorescence-guided surgery will look very much like a photothermal therapy program. So that's one aspect of it. The other aspect of it, they came to were that we would need 300 patients in fluorescence-guided surgery to support safety. But only may need 100, 200, 250 patients to prove efficacy. So that means we get a lot of flexibility in collecting the 300 patients to support safety. And that means actually we can cut the cake, so to speak, in many different ways. We can do 2 regulatory trials. We can do some of the patient on photothermal therapy. We could do it other indications. So we give a lot of flexibility, and photothermal therapy plays into this. So that was a short one, and maybe Andreas, if you want to.
No, I totally agree. So what Morten also said is we didn't ask directly because that came later. So we didn't ask them about photothermal therapy in the interaction, but their feedback on what they wanted us to do in fluorescence-guided surgery kind of makes it easy to include it. It won't cost extra patients because we need extra patients for the safety anyhow. Right.
And then you have a pre-IND meeting coming up with them as well. So is that when you would be addressing?
This was basically the pre-IND meeting, I think you will normally call that, and then it's kind of end of phase 2 trial. So the data we will have another interaction with them.
Yeah,
Right.
we probably will have several interactions with them, most likely. So both on the photothermal therapy, more advanced in fluorescent guided surgery. So there'll probably be a couple of interaction, and it's the normal procedure. We got some feedback, some of the suggestions they have to us. We look at them, we come back with some refinement, and we go down very much into the detail, which is important when talking with this agency and the trials. So I would imagine there will be a couple of interactions as we go on.
Yeah. And what do you think, I mean, is the scope for potentially getting something like a breakthrough designation? What would you need to show to have a good shot at that?
That's also a big, what can I say, an important questions. I think we would like to, before we promise anything, like to see some data on it. But I mean, as a generic comments on it, the more benefit you provide to the patient potentially compared to alternative, the more the likelihood is that you get some kind of accelerated process. And the photothermal therapy has this, what can I say, potential. But let's see some data before we promise on it.
Yeah, and on top of that, the indication, the comparator, what's out there right now is really bad, so or not very good. So there are several reasons why, even just in glioblastoma, but even more once adding photothermal therapy, that we get some kind of the different accelerated mechanisms.
Right. And then in terms of the powering of the study, you mentioned you need, say, somewhere between 100 and 250 patients for the efficacy. What kind of range would that likely give you in terms of powering the study, and what would be the bar for success?
Yeah, I mean, basically, what we will do is that we will the bar would of course be a significant difference in outcome. And outcome can be measured in many ways. Objective, that you look at an MRI, and of course you do a follow-up. It could be a recurrence-free survival, typically in this case.
Yeah.
So how much do you extend that? And typically, I mean, there are some rule of thumbs. You want a couple of months at least. You have to remember that the time here is very short anyhow. The median survival is around 1 year. So here adding 1 month or 2 months is actually a big thing, relatively seen. Then and that's also part of what will be interacted with the agency, because they have opinion on what it takes. And that's where I have the numbers from. Typically, you will always, I mean, 3 months is always very relevant, but it might even be shorter here. And then you can calculate back. Then we will get initial data on how efficient we see the photothermal therapy is, or we estimate what we believe, and then you do the calculation, and then you will arrive somewhere between these numbers. So it will be a specific number we arrive at, but it will be based on these data, a relevant difference obtained, but also what we believe the method will be capable of doing.
Yep.
So it remains to be calculated more precisely.
And then I'm mindful about time, so I think we better move on. But maybe you can talk quickly about the go-to-market strategy you have.
Mm-hmm. Yeah, I mean, the go-to-market, I mean, we have kind of alluded to that before. One of the things we like with aggressive brain cancer is that it's an indication of a size that we can handle ourselves in the US. It's very concentrated, so few customers concentrate on a few places, and they each treat several of the patients. Where when we go to head and neck, lung, and some of the larger indications, it's much more likely to partnering, because we need some more muscles to bring it out to a broad variety of customers spread all over. So this is also kind of the reason why we select these two indications. We have one we can pursue ourselves, and we have one that we can partner and sell out without even selling out some of the major indications still that is to come. Yeah. So we feel that we are very flexible there as well, and decrease the risk of being squeezed into a corner by partnering or things like that. So it's how we put it up.
But does photothermal therapy make it a less attractive option? I mean, given the pricing aspect that you mentioned, where having the equipment calibrated to it, does that make it less attractive to go there yourselves, or say at least without a partner involved?
No, it makes it much more attractive, because what we can do now, it's still the same number of customers, the same number of surgeons, and we can just sell a higher priced product to them. So it makes it much more attractive ourselves.
Yep. And then in terms of the partnering landscape, what are the companies that have equipment that can be used with FG001 in principle?
I mean, it's basically many of the surgical equipment. So, I mean, old days, I mean, you have a knife, basically. Now you get equipment, you get scopes, you can look into the patient, you can do robotic surgery, you can do a smaller, minimal invasive surgery. You have microscope, you can see smaller structures. So everything get more precise and smaller, basically. And what happened is that a lot of this company have added imaging equipment on the surgical equipment, meaning that you can see things as well. And as you have ICG, they have also imaging system that can see in infrared light and see ICG, meaning they can also see our product. So actually, they're already set up for this. And really, what they are missing is, I mean, automatization is a very important thing for them as well. And what they're missing is something that can kind of guide them. And here we are. We can help them. We can light up and we can guide their equipment. So the synergy between is quite important.
And what about, let's say, I mean, you gave a target of partnering initially with someone in some indication already in 2024. What gives you the confidence to put that as an actual target for this year?
I think it does, because what we have seen is that the partner is leaning more forward than they did in the past. So in the past, they have been more reluctant to, because they want to see what happened in this field. Now they see that they started getting some of the structures that is needed to make this field really commercially interesting and get it out to the patients. We see, for instance, in the untargeted investment where Olympus Venture Fund participated, Johnson & Johnson participated. So we start seeing them being actively involved in the field. And it's also the feeling we have when speaking with them. So it's kind of, there's one chair too little, and they now want to move and get sure that they get the dye for their equipment. Is kind of what we see.
Okay. And I guess last question here is before we run out of time. What's the funding situation? What's your runway?
We can run until end of 24. And before we start up the studies, next studies, we would most likely need to funding before that, but that means in third quarter. So we're quite well funded for the plans we have right now, and we have not squeezed in any way. So yes, well positioned.
Okay, so a quick follow up on that though, just what's been the feedback then since the phase 2b on the idea of accelerating the photothermal therapy development from investors?
It's been extremely positive. We have all over. I mean, like you have written yourself in your note, is kind of what we have seen other analysts have of opinion, and it's also generally the feedback we get from investors. So quite positive, as we believe ourselves.
Terrific. I mean, to me, it's a really important thing about what you guys are doing as well, is that you're not just blindly going with, this is our plan. It's you have to adapt to the data that comes up and take the right decision based on that. So I think to me, it's great. So keep doing what you're doing. And thank you for your time. I know we've run out. So thank you all for your attention if you're tuning in.